in your cells, what's happening generally, you're taking glucose and storing it, breaking it down, making ATP. In the process, you're also going to form carbon dioxide and water, right, with the oxygen that you're going to use with the glucose. And so that's what's going on. Glucose floating around your blood, going into the body tissues, and we're going to make ATP somehow. Take the bond energy stored in the glucose bonds, put it onto that phosphate bond. Your muscles, which need a lot of ATP, right, when they're working hard, they need almost more than any other, I guess with the exception of your cardiac muscle, because actually this will work for both what we're talking about, cardiac and skeletal muscle, but let's just focus on skeletal. We have three ways of producing ATP from glucose. Well, not from glucose. These are from glucose. We have three ways of getting ATP, right? Because ATP is floating around, but it gets used up quick. So we got to have a ready store of it or make it. So we're going to go over these three different ways, right? The fact of creatine phosphate, which is going to hold an ATP, and then these two forms of catabolism, right? Breaking down glucose and getting a certain amount of ATP. So first one, creatine phosphate. You may have heard of creatine. So the idea behind it, it's a small molecule. It could go pass through your digestive. It's basically an amino acid. Well, creatine is. It's one of the ones that aren't used actually in protein production. It's a separate one. So it's a very small molecule that won't, it can get through the digestive tract and into your system. So that's the first part you want to know about any supplement. What happens? Can you get it through the digestive tract? And then what happens to it in the liver, right? So for this one, they've shown that there is some significant effect, right? And it doesn't have any side effects like steroids do. But your creatine phosphate, the thing about water, I think they said it maybe absorbs water, it makes your muscle cells hold a little more water possibly. So that's a possibility. But the idea behind it is that your ATP, as I said, needs to be floating around, right? Your sarcolemma and your plasma membrane and your myosin heads ready to be used when you need it. Because if you don't have it, your muscles don't work in so many different ways, right? But when you are relaxed, your muscles keep making ATP through these other mechanisms, which we'll talk about. And when you do that, you don't want to just get rid of that, right? You want to store it some way. And so they take that ATP and they are going to attach it to this creatine molecule to make this creatine phosphate. That is, I'm sorry, they take the ATP, they take the bond and make creatine phosphate out of it in a relaxed muscle. Okay, and they're going to do that through a kinase, an enzyme, right? Creatine kinase. It is going to catalyze the transfer from ATP, this excess, this is going to be excess ATP, right? Because you're constantly making them in preparation for it. And this creatine kinase is going to, cause this relaxation, right? It's going to take that phosphate bond basically and stick it onto that, right? To make this creatine phosphate. So now you have all this excess creatine phosphate comes, right? And so now you got a whole bunch of creatine phosphate and said in a relaxed muscle fiber, that's where most of your ATP is going to be stored. So the idea behind taking, and you have different forms of it. I'm sure you probably take creatine phosphate or creatine, what do they have? Hydrochloric something, slightly different forms, which are more or less effective supposedly, right? But you have all this extra creatine phosphate, right? When you do need it, like when you go to the gym all of a sudden, and you are, going beyond your already cytostolic ATP, now you have excess creatine phosphate. That's the idea. And now you have a little extra energy where you might have got tired before. So that's the idea of taking supplements, creatine phosphate, but it's a natural thing in your body right here. So when contraction starts, right, and all of a sudden ADP levels, because you're breaking down ATP in the process, And now all of a sudden, all your free floating ATP is starting to get used up. Your creatine phosphate could kick in, is available for that same creatine kinase to kick in, take the creatine phosphate, take it from ADP, take the phosphate group, add it to the ADP. Right, takes the phosphate group here, adds it to the ADP. Now you have more ATP floating around, ready to be added to the myosin head or whatever, right? So that's how you're going to generate, right? You store it. The same enzyme is going to be, it's a reversible action, right? You could take creatine, make creatine phosphate. You could take creatine phosphate, turn it back to creatine. That's all just to transfer this phosphate group from or to ADP, right? So these are the two molecules involved, and they're going to be exchanging the bond right there. That's your first source of ATP, right? It's not a very long one, only a couple seconds or 15 seconds or so gives you each molecule. But of course, if you have a lot of them, then you'll have more energy. That's why you want more creatine, I guess, right? So this is your first source of energy, right? It's a very fast reaction that transfer the phosphate group. So this is your first choice to do, right? That's going to be the first thing that's used up. And the more you have in it, presumably, the more you can hold off these other forms where you have to actually make ATP. So that's your creatine. That's the first thing, right? So your creatine is in there. But the fact that there's ATP to make creatine phosphate, asks us, well, where did that ATP come from in the first place? Right. So that's the next couple of things we're going to look at right there. But first, I want to note that for creatine, there is a natural sort of spontaneous breakdown of both phosphocreatine and creatine into a waste product called creatinine, which is very different because it's a waste product, right? This form versus this, right? Basically, it's just a waste product. It just happens. It's part of the natural process that goes on in your body, right? In your muscle. And remind you, we're talking about skeletal muscle and cardiac muscle. Before we get into that, the CK, this is, I said it's cardiac muscle as well, right? There's different forms of creatine enzyme creatine kinase in your heart, in your muscle and stuff. And when you have a muscle damage or a heart attack, right, and you get your muscle cells are damaged, they spill out a lot of this creatine kinase, because otherwise that's something that's not normally in your blood, right? So when you do have some kind of damage like that, they can check your CK levels in your blood to kind of measure the extent of damage right there, or know that something has been damaged inside you right there, right, by levels of this creatine kinase, because it actually busts out of the ruptured muscle cells, right, either cardiac or skeletal, and the particular type will tell you if you have, cardiac muscle damage, right? So that's just one thing I wanted to mention through this CK. All right, but then the second part of it was that there was some waste product based on this metabolism where you had the formation of creatinine, and this was something that's normally processed, right? It goes into your blood and it's going to eventually be filtered through your kidneys. This is something you'll learn about when you get into Bio 145, right? Your kidney takes in all the blood and it gets rid of waste products, right? It's going to filter it in a very complicated process. And it's going to get rid of things like creatine, which is a waste product, as well as things like uric acid and stuff like that, and urea, right? So creatine is a waste product that you normally want to get rid of. And there's a certain amount floating around in your blood, and a certain amount you should be peeing out from your kidneys, right? Getting rid of it. So the reason I'm mentioning this, even though you don't really have to know it, just mental note, this might come up in Bio 45, or it might come up in your life, right? Because it's a common diagnostic tool, creatinine levels to diagnose kidney function, how well your kidneys are working, right? So they can measure the amount of creatinine in your blood, right? And then they could compare to what's in your urine, right? And that will tell you how much your kidney should be getting rid of a certain amount, relative to how much is in your blood. And if it's not, that means you have some kind of renal failure right there. That's just one of those other things that may come up in your everyday life. So that's creatine. And at a certain point, your ATP is used up, your creatine phosphate is going to be depleted if you haven't taken enough supplement. And even if you do, eventually it's going to be used up pretty quick. So now we got to take this glucose that's floating around in your blood over here, right, from digestive systems, floating around in your blood. This is our energy source. And we'll see it's going to get how it's going to get exactly into our blood, which you kind of know already. We're going to get it from the blood, break down glycogen to get glucose, and then we're going to take that glucose and break it down, right? So just again, to kind of remember what we talked about a long time ago. Right? What we talked about was the interstitial fluid and how it served as a medium, right, in between your capillaries, the small blood vessels, where things were coming out like oxygen down its concentration gradient. CO2 is going in, right, through the interstitial fluid, right? So that was that exchange between the body cells and the blood vessels, right, intermediately through the interstitial fluid. So in this case, for right this minute here, we're thinking about nutrients, right? And mainly glucose is coming out of the blood vessels and diffusing through the interstitial fluid and then into the cells, right? So in this case, here the blood, here's a fascicle, here's individual muscle cells, here's the small capillaries that are lying around it. Not shown would be like the endomysium, right? That would provide the sort of interstitial fluid kind of holder, let's say. But stuff was leaking out of here and then going into around the cells, right? And here's the cell membrane over here. So amongst that stuff around here was glucose. And then glucose was going to get inside the cell, especially in response to insulin. There's glucose transporters. We're going to be inserted into the cell membrane. In muscle, they're probably always kind of there, but they can be increased with insulin. And their glucose is going to be transported in, right? Facilitated diffusion. You just need that transport. Glucose will be higher out here than it is in here. So blood glucose goes into the interstitial fluid and then gets into the cell through here. By the way, while we're here in diabetes, right, what happens is the cell becomes insulin resistance, right? It sees insulin, doesn't care about it, right? And it doesn't put glucose transporters in the channel. And then you have all the not enough glucose in here, which makes you really like weak and tired. And you have too much blood glucose, which causes all sorts of problems. When we talk about homeostatic conditions, right? I talked about a balance, right? The right amount, not too much, not too little, right? The homeostatic conditions. With oxygen, right? You're never talking about, oh, there's too much oxygen in your blood, really. And it's the same thing evolutionarily, right? There wasn't really a big problem with too much glucose in your body, right? It was like all about, if you have glucose, take it up. And if there's any excess glucose, you got to store it. So it's always about like getting enough glucose in your body right there, so you can bring it into your cell. There was never like a time when you had so much glucose until now, right? Because now we just have like all the sort of that kind of food around. So we're not really designed to have this much glucose, right? We're designed to like take in glucose and store it as much as possible because it's not like such a plentiful thing. That's just a kind of a side note here. In any case, glucose coming out, getting into your cells through this glucose transport. The other way, very particular, not very particular, but pretty particular to muscle, is the molecule glycogen, right? And glycogen, if we remember, was a whole string of glucose molecules, right? It was the storage form of glucose, right? A whole string of glucose molecules that can be easily broken down, right, into glucose. Right, so you break down, you have all this glycogen, these long strings of glucose, and you can break down that and have a lot of glucose, right? So this is one of the storage things. So glucose is also produced by the breakdown of glycogen in your muscles and your liver. Your muscles and the liver are two of the types of cell that actually make glucose. That is, when you have too much glucose in your blood, you don't need it. You are going to, just like when you have too much ATP, right? You want to store that phosphate and creatine. When you have too much glucose, you want to make those glycogen molecules to store it, right? To keep it ready for action. So hundreds of glucose monomers combine to form the polysaccharide glycogen. And then so you have all this stored capacity within your muscles and in your liver. Your hepatocytes are the other place that does this. So your muscles are kind of unique, and your liver as this being able to make and store glycogen. And most of your glycogen in your body is over here in your skeletal muscle, right? In your total body, there's about, this so much, 375 grams, whatever that is. Somebody could do the calculation here, stored, ready to be kind of broken down, and the glucose can then be broken down, right? All right, so you got coming from the blood, but you also have the storage of glycogen, right? Because we're always going to need, we always want to be prepared and needing glucose, right? Which we're going to use to do what? We're going to break it down now. So we're going to break down those bonds, and next time we'll talk about how the mitochondria break down glucose for ATP using either oxygen, the aerobic pathway, or without oxygen, the anaerobic pathway. All right.